Hello, everyone, and welcome to today's broadcast, Integrating Novel Advances in Gene Delivery and Genome Engineering for Therapeutic Application, presented by Dr. David Schaffer, Professor of Chemical and Biomolecular Engineering, Department of Bioengineering, Director of the Berkeley Stem Cell Center, and University of California, Berkeley. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Schaffer. I will now turn the presentation over to him.
I'd really like to begin by thanking LabRoots for the opportunity to share some of our work with you today. So I'd like to take a historical perspective and talk about the nature of healthcare problems in our country, the United States, over the past 100 years, where a century ago, the leading causes of death in the US were tuberculosis and influenza, which are short-term acute infectious diseases. And with advances in medicine over the past 100 years, namely antibiotics and vaccination and other public health care measures, by and large, a number of these short-term infectious disease problems have been largely addressed within the country. And what this has done has unmasked a large number of other indications that are shown on this slide here. So regardless of which particular tissue, you can take a look at conditions such as Alzheimer's or Parkinson's or rare disorders such as Huntington's or lysosomal storage diseases, where often the progression of these diseases doesn't occur on the timescale of days or weeks. They progress instead on the timescale of years to, in some cases, even decades. So this progressively robs patients of their quality of life, as well as drives up the cost of healthcare within the US. So shown here is a percentage of GDP, the total value of goods and services produced in our country every year, that is spent on healthcare. And this percentage, even though the GDP has been going up on the order of 2 to 3% per year, the fraction of the total GDP that is spent on healthcare has been rising at an even faster rate. So that in 2016, which is the most recent year for which data are available for us from CMMS, we spent 18% of our wealth on healthcare. So to be able to address these problems and improve the quality of health, we need to develop better therapeutics, better drugs. And if we take a look at a very basic biological level of potential levels or potential places in which we could intervene to be able to treat a condition, we all know that information in our body is stored at the level of DNA that's transcribed into RNA and then translated into a protein. And each one of these levels within this hierarchy is potentially a drug target. So the vast majority of pharmaceuticals that we currently have available in the market, small molecules and monoclonal antibodies, target proteins to be able to exert their effects. Small molecules typically bind to an active site within an enzyme. Monoclonal antibodies typically bind to some soluble or cell surface extracellular protein and block or modulate its function. So as we move up one step in this hierarchy, we have RNA. And this includes RNAi or antisense or mRNA being developed by companies such as Alnylam or Ionis or Moderna. And finally, if we move up to the level of DNA, this brings in a number of technologies that have been really the primary focus of my entire career. It's going to be the discussion for today's talk, which include gene therapy, genome editing, and cell therapy. Each one of these operates at the level of DNA, where gene therapy typically adds more DNA to a genome, genome editing modifies or changes the existing DNA. And finally, cell therapy is adding entirely new genomes by adding cell replacement therapies to regenerate a tissue that's been undergoing degeneration, for example. So what's the advantage of operating at the level of DNA? Well, if you take a look at proteins and small molecules, these are typically temporary residents of the body that have half-lives on the order of hours to days, whereas DNA sticks around for the life of a patient. So the potential advantage here is illustrated by taking a look at the timescales of some of the conditions that we described earlier. So from between diagnosis of Parkinson's and ultimately when somebody passes away due to Parkinson's disease, typically that interval is around 16 years. Congestive heart failure, it's around 10 years. Age-related macular degeneration is not a cause necessarily for mortality within patients, but typically by the time patients who are elderly are diagnosed with AMD and when they ultimately pass away is around 10 years again. So compared to these timescales, we have small molecules that typically stay in the body on the order of hours, or perhaps a monoclonal antibody where, if we're lucky, it's on the order of several weeks. There's obviously a huge mismatch between these two timescales, and as a result, these treatments are really treatments. They're not cures for the underlying condition. They only partially improve the quality of life of the patient, and they tend to be quite expensive. So I'd like to contrast that today with gene therapy and genome editing tools, which I'm going to be describing over the next few minutes. And there are two clinical trials. Well, the first one has actually led to a regulatory approval within the United States, and the second is a set of clinical trials for hemophilia B, where now we're out almost 10 years and counting from the initial time in which patients were first dosed in those early phase clinical trials, and they're continuously enjoying very robustly the therapeutic benefit of those treatments. So DNA, because it has the potential to stick around for the life of a patient, provides long-term therapeutic benefit that now matches the timescale of the conditions that patients are suffering from. So just to take a step back, we have gene therapy, which is defined as the delivery of genetic material to the cells of an individual for therapeutic benefit. And the key word that has really been a significant limitation within our field over the past several decades has been delivery. So that whether you're delivering a replacement gene for one that might be defective within a patient, or whether you're delivering a construct that encodes some genome editing tool to modify the DNA within a patient, delivery has been a major barrier where these constructs you would like to administer or transfer into a patient's cells are often very large chunks of DNA compared to the size of a cell, at least relatively speaking for a cell. They're not particularly stable inside the body, at least outside of the nucleus. And as a result, it's been very difficult over the past several decades to deliver enough genetic material to enough cells within a target tissue to really begin to modify disease. So over the past 10 to 20 years, however, investigators have been identifying very promising delivery vehicles, most often based upon viruses, where viruses, of course, carry DNA inside of cells for a living. That's what they do when they infect a cell. And you could potentially trick that virus into delivering a therapeutic piece of DNA rather than delivering its own genome. So in this case, for example, we have Adeno-Associated Virus, or AAV, which is really going to be largely the focus of the remainder of my talk, which has been identified in clinical development as an extremely promising gene delivery vehicle. So it has a number of features associated with it. So it's among the smallest of all viruses, on the order of 5 kilobases of information inside of its genome. It only contains 2 genes that are shown here, REP, which encodes some enzymes that mediate the replication of that viral DNA, and the second, which is CAP, which has been a major focus of my entire career. So CAP encodes some structural proteins that self-assemble to form this hollow soccer ball that has 20-sided symmetry. And once this soccer ball assembles, a copy of the genome ends up snaking its way through a pore on the surface of this capsid in order to load the virus with its payload and create an infectious virus. That resulting virus for AAV, it turns out, is non-pathogenic. It's never been associated with human disease. It's a pretty harmless virus. And a number of natural variants have been identified of the AAV. They have differences in the amino acid composition on the surfaces of these peaks emanating from the soccer ball. And this results in different ways of interacting that virus with host cells and host tissues, and leads to slightly different delivery properties or infectious properties for each one of these viruses. So this virus has evolved over tens of millions of years, a number of ingenious strategies within that soccer ball to carry the payload from point of administration until it reaches the nucleus of a target cell. So for example, once that soccer ball reaches the surface or becomes near the surface of a target cell, it binds to a specific receptor on that cell surface, undergoes endocytosis or internalization, has a mechanism to escape endosomes, another mechanism to undergo nuclear translocation, and then that capsid uncoats and releases the viral genome or viral DNA to be able to undergo the next steps of the infection. So this can be converted into a recombinant gene delivery vehicle by simply taking the 2 viral genes, REP and CAP, cutting them out of the viral genome, pasting them onto a separate piece of DNA, which both liberates space for the insertion of a gene of interest, which then becomes loaded into the middle of the soccer ball, as well as renders that resulting virus replication incompetent within a patient because it no longer has the viral genes that are necessary for replication. And it simply deposits that gene of interest in the nucleus of a target cell so that it can maintain long-term production of a protein or an RNA therapeutic from that DNA. So these resulting vectors are extremely safe. They've been in thousands of patients. And again, just to remind you, greater than 90% of the human population has already actually been exposed to AAV, but it's not a pathogen. It's never been associated with human disease. So we're starting from a very safe starting point. These resulting viruses are somewhat efficient in clinical trials for muscle, liver, lung, brain, and retina. They result in very stable or long-term expression or maintenance of that DNA and expression of therapeutic proteins or RNA from that DNA. There have been a number of advances in making these manufacturable to clinical scale, although there still needs to be a significant amount of progress there. But as a result of all these properties, this virus has now enjoyed efficacy within multiple clinical trials, some of which are shown here. So for example, for hemophilia B in the upper left-hand corner, spinal muscular atrophy in the lower right, and then two diseases or disorders that affect the retina and progressively rob people of their vision, choroideremia in the lower left, and then Leber's congenital amaurosis in the upper right-hand panel. So Leber's congenital amaurosis, or LCA2, was actually the basis last year of an application to the FDA to have an approved product, gene therapy, to be able to treat this condition using an AAV vector by Spark Therapeutics. And this became the very, very first FDA-approved therapy, gene therapy, for a rare disease. So we have a number of positive results within clinical trials for a number of different conditions, and so you get the sense that the field is beginning to build momentum. However, for one reason or another, each one of these conditions for which there has been success has been a quote-unquote low-hanging fruit on the tree. For one reason or another, these have been special cases where the natural versions of these viruses have been just good enough to be able to get efficacy. And even so, investigators have had to brute force the delivery, use extremely high dosages or very invasive routes of administration to place the vector right next to the cell target inside the body. So in general, gene therapy is beginning to work, and genome editing can build upon that success, but the majority of human disease targets are still beyond the reach or the capabilities of current gene delivery technology, and we quite simply need better viruses. So what are the current limitations with some of the natural versions of AAV? Well, I alluded to one of those, which is the fact that the majority of us have already been naturally infected accidentally by AAV, and we've been, as a result, immunized against AAV, been vaccinated. So we have high concentrations typically of antibodies that are waiting to neutralize AAV, whether it's a natural AAV infection or an administration of a medicine, a DNA therapy, based upon AAV. Even if the virus is able to make it past those antibodies, and it turns out those antibodies are a pretty significant challenge, it's the most common reason that patients are turned away from a clinical trial. But even if the virus does not have antibodies as a barrier, it often biodistributes to the wrong tissues inside the body, has inefficient spread when it reaches the surface of those target tissues, has zero capacity to target one cell type versus another within those tissues, and even if it reaches the surface of a target cell, most cells in the body are not highly efficiently infected by AAV. So we came into this field around 20 years ago and began to observe some of these challenges and hypothesize they arise from a very simple kind of no-brainer consideration, which is that nature never evolved viruses for our convenience to use as medicines. So if we want to optimize these viruses for therapeutic gene delivery, we need to re-engineer them at the molecular level to make those soccer balls better as therapeutic pharmaceutical gene delivery carriers, rather than being at the mercy of whatever natural viruses nature happens to have thrown over the fence at us. So when we began this work, we began to recognize and embrace the idea that evolution is what created these viruses in the first place. So why not simply continue that process of evolution, but change the direction so that we can, in a laboratory setting, evolve the viruses towards the properties that we need them to have to be therapeutic gene delivery vehicles, again, rather than being at the mercy of the natural versions of these viruses. So we developed an approach, a platform technology to be able to do this, which is described in this slide. So the soccer ball is encoded by the CAP gene, and so we have the potential to engineer that CAP gene to improve the properties of that soccer ball delivery vehicle. So the way that we do this is that we take these sequences of these CAP genes, and I mentioned to you there are a number of natural versions of AAVs. Perhaps we take not just one, but several different versions of these CAP genes, And we continue the process of viral evolution to make better versions for our therapeutic applications. So all evolution involves two steps: genetic diversification to create a rich gene pool, and then Darwinian selection to the fittest. So we have used 37 different approaches to create 37 individual libraries of the cap gene in which we've introduced diversity into that capsid in a variety of different ways. We then take this very, very large pool, which in sum total amongst those 37 libraries that we simply pool all together to create a master library, we have several hundred million variants of AAV, which we think compares quite favorably to around the half dozen AAVs that are currently used within the clinic. We then package these AAV genomes into a large number of particles so that we now have a library of particles where every single variant protein shell or variant soccer ball contains inside of it the DNA that encodes that soccer ball. So we can then select this very large pool for the fittest variants that, for example, are optimal at being able to mediate delivery to the liver or to the brain or to the lung or to the skeletal muscle. And we can screen or select them. I'll show you a couple of examples to be able to isolate the fittest from this pool and simply spin around this loop multiple times so that we progressively improve the properties of this virus. And at the end of that evolutionary process, or perhaps at certain junctures along the way, we can pop open those particles, sequence the cap gene, and gain insights into the process of evolution as this very large, several hundred million variant library converges now down just to several lead variants that have the properties that we would like them to have. So we have been doing this now for 20 years and have evolved a number of different highly optimized targeted viruses to be able to address a number of those problems I mentioned to you earlier for a number of cell and disease targets. And I'm going to give you a couple of examples today within the retina, as well as in the brain and spinal cord. And then I'll talk about our efforts to translate to the clinic. And finally, just show you several slides on a new tool that we've developed recently to be able to accelerate the process of genetic diversification and selection. So first, the retina. So the retina, as based upon that FDA-approved product to treat this blinding disease, Leber congenital amaurosis, is a very important and promising target for gene therapy. So the structure of the retina is that on the back surface of our eye, we have painted several layers of neurons. So for example, this ONL, or outer nuclear layer, contains the photoreceptors that sense photons. This information, once it activates those neurons, is translated through several interneuronal layers until it reaches the retinal ganglion cells within the ganglion cell layer. And this information is sent through their axons, which comprise the optic nerve, into the brain and for processing of that visual information. So one particularly important area within the retina is within the central part of the eye called the fovea, or the macula. And this contains a really high concentration of cone photoreceptors, and these are important for high acuity vision. So if you're reading across a page, the reason your eye moves back and forth is so that you can keep your fovea focused on a region where you need the highest visual acuity. Unfortunately, mutations in over 200 genes can result in the degeneration of photoreceptors within the eye. In particular, loss of cones is something that's particularly devastating to patients because that's leads to loss of their high acuity or high accuracy vision. So it'd be very advantageous to deliver a therapeutic chunk of DNA to the cells in the outer nuclear layer, either the photoreceptors or the underlying retinal pigment epithelium, to be able to treat these genetic disorders. And furthermore, gene therapy could potentially be harnessed to treat other complex diseases within the eye, such as macular degeneration. So the most advantageous place to deliver or to administer a therapeutic would be into the vitreous fluid of the eye, so-called intravitreal injection. And this is very, very safe and simple in its administration. It's actually done several million times a year within the United States to treat patients with age-related macular degeneration with drugs that are developed by Genentech and Regeneron, for example. So this has the advantage that it's a very simple outpatient procedure. It takes an ophthalmologist around 4 seconds to do an intravitreal injection. In addition, once the therapeutic is within the liquid humor of the eye, it has the potential to come into contact with the full surface area of the retina, and as a result, in theory, protect all the cells, all the photoreceptors within the retina. Unfortunately, AAV, which is around a 25 nanometer particle, faces a number of barriers in reaching those target cells, reaching those photoreceptors after an injection into the vitreous. So specifically, there's several hundred microns of dense fibrous tissue which separate that vitreous fluid from the outer nuclear layer, which contains the target photoreceptors. And the natural versions of AAV can't penetrate through that dense tissue in order to reach the target cell. And this is really representative of a number of problems in gene delivery where it's hard to get viruses to very difficult to reach remote locations inside of tissues, or difficult to get viruses to penetrate throughout a tissue. As a result of this limitation, the clinical trials that have been conducted to date, including the ones for LCA2, which led to that FDA approval, have involved a subretinal surgery where the needle is punctured through the retina and fluid containing the virus is injected underneath the photoreceptors, which causes a retinal detachment. The fluid forces its way underneath the photoreceptors, which separates the photoreceptor layer, or separates the retina from the underlying tissue. And in patients who are undergoing a retinal degeneration, you might imagine that that adds insult to injury. It further damages the tissue. And in addition, within this fundus image, which is what an optometrist sees if they're looking through the lens at the back of your eye, a subretinal injection places a bleb of fluid within only a small fraction or percentage of the surface area of the retina, on the order of 10 to 15%. So if the therapy only reaches the cells that are in contact with that fluid at that injection site, it really sacrifices on the order of 85 to 90% of the other cells across the entire retina to the disease. So in that FDA-approved gene therapy, the majority of the retina undergoes degeneration, only a small fraction of it, as a result, is rescued. So we asked the question, can we engineer AAV to do this, to make the trip from the vitreous fluid all the way back to the photoreceptors? So because there's no cell culture model that can really capture the complexity of the retina, we began to do this with an animal model. And the way that it works is that we take our entire pool of several hundred million AAV variants, put them all into a single syringe, inject them into the vitreous fluid of the eye, wait a couple of days in order for those variants to reach the target cell, the photoreceptors, harvest the photoreceptors, isolate the AAV genomes, the cap genes that made it to that target cell, amplify those, repackage them, and spin around this loop, re-administering this pool to the animal to reach the photoreceptors round after round, so that we end up with convergence from on the order of 10 to the 7th or 10 to the 8th variants of the AAV down to just several candidate variants that have now been heavily selected by evolution for having the properties that we want them to have, namely the ability to infect a photoreceptor from the vitreous. So in this initial work, we did it as a proof of concept within a rodent model, a mouse, and we found that this tens of millions or 100 million variant pool converged down to just a small handful of variants, shown in a pie chart here on the lower left-hand side, where actually the dominant variant comprised around 2/3 to 3/4 of the entire pool. So there's a cartoon of that, the crystal structure, the capsid structure of that variant in the lower right-hand corner, and you can see that there were some changes shown in green into some key loops on the surface of the virus, which changed the way it interacted with tissues and with cells, and potentially made modifications that enabled that virus to be able to reach the target cells. So shown here on the left-hand side is a slice of tissue where the bottom part is the vitreous fluid, the top part is going to be the photoreceptors, and this is an intravitreal injection of AAV2, which is the variant that is used within that FDA-approved product. So you can see exactly why they have to do a subretinal injection, because if you administer AAV into the vitreous, very little of it reaches the photoreceptors. So if we take the identical dosage of our engineered variant and administer it to the eye, we can see that we're lighting up the entire retina, every single cell layer, including, importantly, the photoreceptors. And furthermore, this delivery is not limited to 10% of the surface area of the retina, because we're putting it into the vitreous and the virus can diffuse across the entire surface area of the retina. We're hitting the entire retina. So we began to use this to be able to treat an animal model of a human disease. This is a blinding disease called retinoschisis, where loss of functions or mutations within a particular gene called retinoschisin results in degeneration of the retina and the formation of these cavities or schises in the eye, as shown in this image, this cross-sectional image of an 11-year-old boy who's progressively going blind. This protein is naturally produced by photoreceptors, and as a result, you need to deliver the gene encoding this protein to the photoreceptors in order to repair the condition. So shown here is the animal model. This is a wild-type animal. This is a retinoschisin animal, and you can see that it copies or phenocopies the human disease quite well with the formation of these cavities or schises. And if we take the engineered virus, which is called 7M8, and load it with an inert gene, including the green fluorescent protein, you can see it doesn't modify or change the formation of these species. But if we load the 7M8 engineered virus with the therapeutic DNA encoding retinoschisin, we can see that there's structural rescue of the tissue, a preservation or maintenance of the full thickness of the retina, And finally, within a ERG or electroretinogram, you can see here a wild type or natural ERG shown with the dotted line in the upper panel. A retinoschisan animal is shown here with the green the green profile, where this animal is progressively going flatline or blind. And the middle one, which is the dark color, we can see that we're progressively improving the capacity of these animals to see or to exhibit ERG behavior. And this preservation, or the degree of it, is maintained throughout the entire life of the animals, shown in the bottom left, where only in the case of the engineered virus delivering the retinoschisan gene do we have preservation of ERG responses for many months on end. And we see that a number of negative controls, which include natural versions of AAV carrying the therapeutic DNA, or the engineered version of AAV, R7M8, carrying an inert gene, GFP, which all overlap with control untreated animals. So I'd like to move on to another tissue target, which is going to be the brain and spinal cord. And this is another tissue or condition, or tissue where there are a number of conditions which represent serious unmet medical need. Anything from Alzheimer's to Parkinson's to rare monogenic disorders such as Huntington's disease or amyotrophic lateral sclerosis. So the problem for delivery to these tissues is that they're very large tissues. The brain is large in volume, and the spinal cord is very long. And it's very difficult to treat them like a pincushion, meaning it's hard to do enough direct injections into these tissues to be able to expose the full volume of those tissues to a virus. And this is illustrated by a recent clinical trial at Institut Pasteur, where to be able to treat a condition called Sanfilippo B, which is a lysosomal storage disease, investigators actually drilled 8 holes into the skull of patients and it did 16 injections. And this was sufficient to restore around 14 to 17% of natural levels of the target gene, which is an enzyme in this disease. And this was not sufficient to be able to significantly modify the disease. So they had to go through heroic efforts to be able to get enough virus into the brain through a large volume of the brain, and this was still not sufficient to be able to change disease progression. So a number of years ago, we noticed another mechanism, or observed another mechanism whereby virus could spread inside the brain. And this is called retrograde transport, where neurons, of course, have a cell body and then they have an axon, a process. So if you deposit the virus or inject it right near the tip of that axon, it can undergo endocytosis or internalization and retrograde transport back to the cell body. And this provides a mechanism where you can spread the virus through a large volume. For example, if you have a large number of neurons, which all send their projections of their axons to just one location, by doing a viral injection at that location, you can get the virus to spread through a large volume. And another example was that in a subsequent study, in this case, Brian Kaspar, the lead investigator in the study, did an injection into muscle of the virus, and it was retrogradely transported along motor neurons back to the spinal cord. And this was used as a way to deliver a neuroprotective factor to preserve the viability of motor neurons within the spinal cord in a mouse model of amyotrophic lateral sclerosis. So this is extremely promising as a way to remotely deliver DNA to a large numbers of cells and get it to spread within a target tissue. But the problem was this technology was licensed by a company that began to treat mice and then rats and then non-human primates with AAVs, and found that the natural AAVs did not undergo sufficiently high levels of retrograde transport for this to be practical for human clinical use. And another way of visualizing this problem is that it's very likely that in its tens of millions of years of evolutionary history, natural AAVs have ever been rewarded evolutionarily for the capacity to retrogradely transport within a neuron. AAV is a respiratory virus. So we decided to set up our own evolution to reward retrograde transporters. And the way that this worked is that there's a location deep within the brain called the pons, and there's several layers of the cortex, both within rodent models as well as within humans, that send their projections to the pons. So we injected the entire library within the pons, waited a couple of weeks, harvested the cortex, amplified the viral DNA that made it there, repackaged it, re-injected it into the pons, and spun around that loop around a half dozen times, so that again, the 100 million variant plus library converged down to just a small handful of candidates. And specifically, there was one dominant virus. We then took this one, loaded it with a reporter gene, specifically that led to the expression of TD tomato, a fluorescent protein, injected it into the pons, and you can see here with a single injection of this virus, we're able to light up the entire cortex. So we wanted to quantify the efficiency of this process, and so this is a cytosolically soluble version of a fluorescent protein. So to quantify, we actually used a nuclearly localized GFP so we can count individual cells stereologically. And you can see here, this is AAV2, a natural version of AAV. There's an injection in the lower left-hand corner. You can see a cluster of green cells. However, there's nothing in the cortex, which is a long strip of tissue in the upper right-hand corner. This is an engineered version of AAV that is developed by another lab in the field. Injection into the pons maybe leads to a few speckles of cells in the cortex. And this is our engineered version of AAV, where with a single injection in the pons, you can see that we're spray painting the cortex with very large numbers of transduced nuclei, meaning we're getting very efficient expression. So if we quantify this degree of retrograde transport, AAV1 through AAV9 are very low. Canine adenovirus, or CAV2, is actually used in neuroscience as a retrogradely transporting virus to be able to do mapping of pathways inside the brain. It's not particularly efficient at doing that. And we find that our engineered version, which is called AAV2 retro, is actually 50 to 100-fold more efficient at undergoing retrograde transport compared to natural versions of AAV. So for the remainder of the talk, I'd like to kind of transition into using these tools as a way to deliver genome editing capacity in vivo. And we use this, really quickly, I'm going to show you some spread of this virus. We use this virus, this AAV2 retro, to be able to get very strong spread of constructs inside the brain. So in this image, we ask the question, by choosing several strategic locations for doing an injection within the brain, can we actually maximize spread along a number of different axonal pathways to get really, truly strong spread throughout the entire brain? And so in this case, we used a red, a green, and a blue fluorescent protein encoding viral genome, did 3 injections, and you can see with just 3 injections, you can light up the majority of the brain. So we then loaded this virus with a CRISPR-Cas9 construct, and specifically, we utilized an animal that expresses now TD tomato naturally at high levels within the cortex. And then we engineered a version of Staph aureus SA-Cas9 that was armed with a guide RNA that enabled it to cut near the 5' end of TD tomato. And as a result, through the introduction of insertions and deletions during non-homologous end joining, it should knock out TD tomato expression within the cortex. So shown here in the right-hand panel, we quantified the fraction of cells within the cortex that were transduced or infected by the virus after injection into the pons and retrograde transport of the viral DNA to the cortex. We quantified what fraction of those had TD tomato knocked out. So if we used a control construct, which is the left purple bar, this is a control construct that contains a non-targeted Cas9. You can see that the majority of the cells, greater than 95%, are expressing TD tomato. But when we use a guide RNA that arms the Cas9 to be able to attack the TD tomato gene that's integrated into the DNA or the genomes of these animals, you can see that we're knocking out 80 to 90% of expression of TD tomato within the cells that were infected. So I'd like to now talk about application of the delivery technologies to be able to carry therapeutic payloads, therapeutic genome editing capacity into cells within the nervous system. This, of course, could in theory be used using any engineered virus that we created that is highly optimized and is 2 to 3 orders of magnitude more efficient at carrying DNA into any target cell that we might be interested in engineering a virus for. This could be used to carry genome editing capacity into other cells or tissues in the body. But right now, I'd like to talk about amyotrophic lateral sclerosis, or Lou Gehrig's disease, which involves the loss of motor neurons within the spinal cord, brainstem, and motor cortex, and leads to, within 3 to 5 years of onset, paralysis and ultimately death within patients. So around 10% of ALS cases are familial, meaning that the blame for this condition can be attributed to mutations within specific inherited genes within the genomes of those patients. And of the familial cases, 20% of those are due to mutations within a gene called superoxide dismutase 1, or SOD1. And these are inherited in autosomal dominant fashion, so that you can't treat the disease necessarily by adding DNA to the genome. What you really want to do is to erase DNA. You want to knock out a gene. And currently, unfortunately, there are no effective therapies for ALS. So we engineered versions of SA-Cas9, or specifically guide RNAs for SA-Cas9, that armed it to be able to cut the human SOD1 gene. And you can see here in lanes 1, 2, and 3, we have several that are very effective guide RNAs that convert SA-Cas9 to a strong nuclease to cut the human SOD1 locus. We then loaded this SA-Cas9 construct with its guide RNA into AAV, administered it into animals using an engineered version of AAV, animal models of ALS that harbor a mutant version of the human SOD1 gene. And you can see here that this disrupted significantly SOD1 expression within these animals. So shown in the left-hand panel, this is a control construct in which Cas9 has a control guide RNA that's directed against an irrelevant target within the genome. It's not directed against SOD1. And in yellow, you can see there's very high levels of expression of SOD1 within motor neurons, which are shown in red. However, when we arm Cas9 to be able to attack the human SOD1, you can see there's a significant knockdown in yellow immunoreactivity, a knockdown in SOD1 expression. And this is accompanied by the observation of insertions and deletions at the SOD1 locus, indicating that Cas9 cut that locus and non-homologous end joining incorrectly repaired the locus so that it ended up knocking the SOD1 out of its reading frame and ablated expression of this disease-causing allele of SOD1. I'll finally mention as well that the version of AAV that we used is actually more targeted towards motor neurons. It is known that expression of the mutant SOD1 within other cells in the spinal cord, specifically within astrocytes and oligodendrocyte precursor cells, also contributes to pathogenesis or the pathology of this condition. But we wanted to see initially, using this proof of concept virus that's more targeted towards motor neurons, if we could see some extension of lifespan within these animals. And then perhaps we could tackle the astrocyte and oligodendrocyte precursor problems in next generation therapies. So we see here in the upper left-hand panels, A and B, that there's a significant delay in the onset of paralysis within these animals that are treated with the Cas9 directed against the SOD1 gene. In the upper right-hand side, there's a significant extension in the survival of these animals, so that we delay disease onset by around 34% and extended survival by 25%. So we're now using improved versions of the AAV that target delivery not just to the motor neurons, but also towards glia, towards astrocytes and oligodendrocyte precursor cells. And it's our hope that these new and improved versions of the virus, next generation versions, are going to be able to knock out this gene within other cells that contribute to disease progression, resulting in even further extensions in lifespan. So over the next 10 minutes, I'd like to talk about two final steps. The first of these is our efforts to translate into the clinic. So around 4 years ago, also by way of disclosure of financial interests, since I'm a co-founder of this company, we spun a company out of Berkeley called 4D Molecular Therapeutics. And 4D has been developing next generation AAVs for a number of different targets, which I'll talk about in a second. And we've been progressing these into the clinic, both internally within our own programs, as well as through partnership with companies like Roche, Pfizer, and AstraZeneca. So evolution, or nature, teaches you that within evolution, you get what you select for. And I showed you a couple of examples in which we did the selection, in which we did the evolution within a rodent model. So as I'll show you in a second, human beings or primates aren't rodents, and as a result, a vector that's optimized for use within a rodent may not be yet optimal for human clinical translation. So within 4D, we've been doing the evolution for a number of different tissues, and we've been doing it within non-human primate models and human organotypic cultures, with the idea that these are the systems, the preclinical systems, that are as close to humans as is possible. So we want to do the evolution and do the selections within a setting that we believe is going to make the resulting viral technologies directly translatable into the clinic. So we've been doing it and have a number of engineered variants of AAV within target tissues, including the retina, central nervous system, lung, liver, heart, muscle, as well as other targets. And today I want to talk about the retina. So why might a rodent vector not be optimized for use within a larger mammal, and specifically a primate? So first of all, there are differences in the anatomy. This is drawn to relative scale, where the mouse eye is pretty tiny compared to the human eye. And there's on the order of 100 microliters of vitreous fluid within the mouse eye. We have around 4,000 microliters of vitreous fluid in the human eye. So there's a difference in scale. And in addition, there's a difference in the anatomy of the retina, where I described to you earlier that we have this region called the fovea with high concentrations of cone photoreceptors. And mice don't even have a fovea. So if you want to study a cone degenerative disorder, such as macular degeneration, it's difficult to do that within a mouse. And furthermore, there's a barrier within the tissue that lies between the vitreous fluid and the retina. This is called the inner limiting membrane, or ILM. And this is a barrier to AAV delivery into the retina. And this barrier, this ILM, is quite simply thicker within humans and monkeys. So we began to use this rodent AAV just to test whether or not it could work within non-human primate retina. And shown here in the lower right-hand corner, so these are natural versions of AAV within non-human primate retina. They're not great. This is the engineered version. Due to limitations of time, I won't describe for you why we have this particular pattern, but you can see the majority of the surface area of the retina is not protected with this rodent AAV. So we reperformed the directed evolution from scratch within a non-human primate and created a highly optimized version of AAV that should now work within NHP. And due to the similarity in the non-human primate retina to that in the human retina, we believe that this is going to be translatable in the clinic. So to show you results from this, these are four images from non-human primates from an in-life study in which we loaded this optimized soccer ball, this optimized AAV, with DNA encoding the green fluorescent protein. And you can see here at 6 months of age, this has lit up the retina of these animals. We've quite literally done thousands and thousands of tissue sections and stained and examined and quantified the expression. And we see very high levels of gene expression within photoreceptors as well as the retinal pigment epithelium within these animals. So our initial target is going to be a blinding disease called choroideremia. And this is due to mutations within the REP1 gene, which encodes a protein that's important for vesicular trafficking, and for reasons that are not fully understood, compromises the retinal pigment epithelium within the eye, which in turn leads to death of photoreceptors and progressive blindness within these patients. So we'll be initiating a human clinical trial to treat choroideremia very early in Q1 next year. So I'd like to close with describing work that we've done to create kind of a next generation technology to accelerate the process of evolution of viruses, or in principle, any gene. And this is, again, based upon CRISPR-Cas9, which we keep finding as a terrific tool within the toolbox to create a number of different downstream technologies. And this is work that was published about a month ago or so. So I'm going to describe for you the molecule and then talk about the application. So imagine you have a version of Cas9, in which one of the two nuclease sites has been mutated, so it functions as a nickase that only introduces cuts into one strand of the DNA. And we take this nickase version of Cas9 and fuse it to a DNA polymerase, and this DNA polymerase can have some properties of interest, which I'll describe in a second. So upon introduction of this into the cell, along with a guide RNA, it leads to that Cas9 getting recruited to the locus that's specified by that guide. The Cas9 nicks the DNA, this version of this molecule, which we call a volver, then dissociates from that nick, flips around, so the DNA polymerase binds to that nick, and we choose DNA polymerases that are actually nick-translating DNA polymerases. And this will end up displacing one strand of DNA and laying down a new track of nucleotides. And we can choose this polymerase, and then finally, this dissociates at some random time from the DNA, and that flap that's been displaced ends up getting cut off, and then as a result, you've replaced that original DNA with a new stretch of DNA. So the reason that this is of interest is that you can introduce into that DNA polymerase, because it's a very modular system, any number of properties. We can change the fidelity of that polymerase, where, for example, we can use an error-prone DNA polymerase where you can introduce at some defined rate mutations into that strand, that target strand of DNA. In addition, you can change the misincorporation bias specifically so that certain mutations might be more or less likely. You know, A to T might be more likely than a T to A or G to C, or perhaps you want even probability of mutations from any base to any base. And finally, you can change the processivity, which ends up changing the number of nucleotides or the window over which mutagenesis is introduced into a target locus in the genome. So we've used this system and examined it using a number of different approaches, including next generation sequencing. And you can see here on the x-axis, this is a distance from the target site from the nick, and on the y-axis is the error rate, or the rate of mutagenesis at that locus. And you can see that for this particular polymerase, we end up with mutagenesis stretching on the order of 50 nucleotides. We've seen it as long as 250, depending upon the processivity of the polymerase we use. And we end up with a substitution frequency on the order of 0.1 to 1%, so a very high mutagenesis rate. And this is initially done within bacteria. We've also gotten it to work within mammalian cells. And this is also compared to any number of negative controls, which include no mutagenesis if you use an off-target guide RNA, no mutagenesis if you use the on-target guide RNA, but use a nicking Cas9 that doesn't contain the DNA polymerase fused to it. And in sum total, the mutation rate is around 8 million-fold higher than wild-type cells at that same locus that's targeted by Evolver. So we've now been using this to evolve DNA inside of cells. And this can be done in situations where you introduce exogenous DNA into a cell, let's say some target protein, such as a blue fluorescent protein that you'd like to evolve into a green fluorescent protein, just as in a proof of concept problem. So we've seen that if we target Evolver to this GFP, we end up with mutagenesis and recovery of GFP. We're switching from blue fluorescence to green fluorescence, and you can sort that out using FACS. We've also done this evolution on endogenous loci in the genome, where in this case, we're mutating a ribosomal protein to end up conferring resistance to an antibiotic spectinomycin inside of cells. And you can see that we end up with acquisition of antibiotic resistance as a result of evolving this target at rates that are multiple orders of magnitude, 4 to 5 orders of magnitude higher than spontaneous generation of antibiotic resistance. And as a final piece, it turns out you can also do this, take advantage of the multiplexing of CRISPR-Cas9 and introduce 2 guide RNAs, one targeted to one ribosomal gene, the 30S ribosomal protein S5, which is a target of the antibiotic spectinomycin, and a second targeted against 30S ribosomal protein S3, which is a target of streptomycin. And only when you have both of these guides present, and as a result, confer upon those cells the capacity to evolve resistance to both of those antibiotics, only in the presence of both of those guides do you end up with antibiotic-resistant bacteria. So we're currently translating this technology into mammalian cells to evolve things that might be of interest, such as gene delivery vehicles. So I'd like to close with a few bullet points. The first is gene therapy is working. There are now a number of absolutely bona fide clinical successes. And this has been enabled by and large with viral gene delivery vehicles, but of course, nature never designed or evolved or optimized these viruses to serve as human medicines. Fortunately, technologies such as directed evolution can be brought to the rescue to be able to create highly optimized, efficient, targeted versions of these AAVs, in other words, designer viruses that can mediate delivery to any cell or tissue target in the body. And we've used this approach to fundamentally reprogram the properties of these viruses to increase their efficiency, target delivery, and in work I didn't talk about today, enhance the capacity to evade the immune system. We're currently translating this into human clinical trials, as well as working on next generation tools such as Evolver to be able to, in principle, evolve any target DNA inside of a cell, including, for example, a capsid. So I'd like to close by acknowledging the people who actually did this work. The work in the mouse retina was done by Leah Byrne, Denise Dalcara, and Tim Day in collaboration with John Flannery. The work on retrograde transport was the product of Tom Guy, Bamyul Wang, in collaboration with investigators from Genelia Farm. The non-human primate retinal variant was engineered or developed, as I mentioned, at 4D Molecular Therapeutics, with major contributions from our CEO, David Kern, who's also my co-founder, our Director of Discovery, Melissa Cotterman, SVP of Clinical and Translational Research, Peter Francis, as well as other important contributors. And finally, the work on Evolver was the product of Shaked Halperin in collaboration with John Duber at UC Berkeley. So thanks so much for your time, and I'd be happy to address any questions.
Thank you, Dr. Schaffer, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Now let's get started. Our first question is, what other tissues have you engineered AAV for?
So I showed one slide which and very quickly showed some of the target tissues that we've developed technologies for delivery, highly optimized vectors for within non-human primates, and this is work done at 4D. So in principle, this is a broad technology platform where if you can design a selection, such as the ability to reach any target cell within the body, you can use this as a way to evolve an AAV that's optimized for being able to reach that target cell. We've specifically implemented it in the retina, which I showed you data for, within the brain and spinal cord, for liver, skeletal muscle, heart muscle, lung, joint, as well as a couple of other tissues. And the lead program that we're currently developing for the clinic is within the retina. But we anticipate also being able to take in a virus highly optimized for skeletal muscle and for cardiac muscle as the next candidate to be able to proceed into human clinical development.
And how have you addressed the problems of neutralizing antibodies against AAV?
Yeah, I mentioned this issue that the majority of us have just been accidentally infected with natural versions of AAV, and as a result, vaccinated or immunized against those natural AAVs, and we have high concentrations of antibodies directed against those natural AAVs floating around in our bloodstreams as well as in other bodily tissues. So if delivering AAV to a given tissue requires it to be administered into some bodily fluid that has antibodies, and you're using a natural version of AAV, it just means that a fraction of the population is not going to be a candidate for the therapy. And that can be anywhere in the literature from 40 to as much as 90% of the human population is simply not going to benefit from a product that's developed from some of these natural versions of AAV. So we did evolution again and took our entire very large pool and evolved this pool for the capacity to evade pre-existing antibodies that are common within the human population. So specifically, we took the pooled IgG, the pooled antibody fraction from around 1,000 donors, a product called intravenous immunoglobulin, which as a result has lots of antibodies direct against AAV because the majority of those donors were naturally infected with AAV. So we took this, mixed it together with the library, administered it to cells. The virus that was not bound or tightly neutralized by these antibodies, the virus is able to infect those cells. We rewarded that virus and provided it with the opportunity to replicate its genome to increase its frequency within the gene pool, and then took it, re-exposed it to antibodies and spun around that loop so that we ended up evolving now variants of AAV that are not neutralized to nearly as great an extent compared to natural versions of AAV. So we can then take these and kind of view them as lead compounds and further genetically diversify them and engineer them for now the capacity to reach a target cell in the body so that we end up with the best of both worlds, a variant which is not going to be neutralized by the majority of pre-existing antibodies and then does have the delivery properties, the targetability and the efficiency that we want it to have to be able to reach a given tissue.
All right, and looks like we have time for one more question. How could you address the problem that AAV would provide long-term expression of Cas9 when likely what you want is transient activity?
Sure. Yeah. So I spent some time talking today about gene therapy as a way to augment the genome, to add DNA to cells. And this is relevant in situations where somebody has some loss of a function or loss of a gene due to mutation that we simply need to replace that DNA. In that case, you want that DNA to stick around for very long periods of time for the life of the patient. In the case of editing DNA or editing a gene within the genome by delivering a genome editing capability, such as CRISPR-Cas9, you really don't want it to be around for long periods of time. You just want it to be there transiently. You want it to do its job on the genome and then go away, because if you leave that Cas9 around for long periods of time, eventually it can start possibly hitting locations in the genome, off-target locations that could cause toxicity within that cell. And certainly, Cas9 is a bacterial protein. Eventually, the immune system is going to notice that a given cell inside the body is expressing this bacterial protein and might start mounting an immune response against that therapeutically treated cell. So you really want that Cas9 to be there quickly and then go away. So we and others have been developing negative feedback systems where you can take the target site for the Cas9, which is present within a target gene in the genome, and take little copies of it and also stick it inside the AAV genome. So once Cas9 is expressed, it hits its target site within the genome, but also loops around or boomerangs back and cuts the viral DNA that encodes the Cas9, and as a result, eliminates itself. So this negative feedback loop, or this suicide kind of system, provides a pulse of Cas9 activity long enough for it to be able to do its job in the genome, but then it goes away.
Well, I would like to once again thank Dr. Schaffer for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2019. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.